Welcome back to Biomandscape. Today we are going to understand the stages of germination. Seed germination. First, what happens? The imbibition. The seed rapidly absorbs the water from its environment, causing it to swell and soften the seed coat. Then the enzyme activity takes place where the water triggers the release of the gibberellin hormone, which activate the enzyme like alpha amylase to break down the stored food, that is starch and protein, into usable sugar. Then the embryo begins aerobic respiration. It consumes the oxygen to produce the energy required for cell division and growth. And then the radicle emerges. The first structure part of it breaks through the seed coat in a radicle, that is embryonic root, which grow downwards to anchor the plant and absorb the water. And the shoot is forming. The embryonic shoot, it grows upward to the light source, that is a plumule, and then once the true leaf that emerges and start photosynthesis, and the plant become independent of the seed storage of the food. And the essential conditions are: there is necessity of the water, oxygen is required for respiration, soil that is too waterlogged can drown, and the seed being depriving into the air, and the temperature, most of the seed germinate optimal between 20 to 30 degrees Celsius or 68 to 86 Fahrenheit. Through some species have specialized requirement for cold and heat and the light, which is not always required, but some photoblastic seed need light to trigger the process, which other require in the darkness. So the common germination types are epigale, the embryonic stem, the hypocotyl elongates pulling the seed leaves, cotyledons above the ground, example beans, and hypogale, that is the cotyledon remain below the soil surface, and the upper stem elongate like in pea and corn. So for knowing such concepts, follow Biomandscape.